Today we will be explaining the process of meiosis for the EdXLB A-Level Biology syllabus. So first of all, what is meiosis? Meiosis is a type of cell division used in sexual reproduction. It produces 4 genetically different haploid cells. In this video, we will be explaining the steps of meiosis as well as how the process increases genetic variation within a population. Lastly, we will go through a typical exam question. The first step in meiosis is prophase 1. The chromosomes have been replicated, so the cell is diploid. The nuclear membrane breaks down and spindle fibers start to form in the cytoplasm. Homologous chromosomes also pair up, forming tetrads. Crossing over also occurs, which we will explain on the next slide. So what is crossing over? As mentioned on the last slide, crossing over occurs during prophase 1. A chromatid from each chromosome crosses over another and breaks at the chiasmata. The chromosomes are then recombined, leading to increased genetic variation. The next stage of meiosis is metaphase 1. The chromosomes are now attached to the spindle fibers by their centromeres. Each homologous pair is attached to a different pole. During this stage of meiosis, independent assortment of chromosomes occurs. Independent assortment is the formation of random chromosome combinations, which, alongside crossing over, also increases genetic variation. The third stage of meiosis is anaphase 1. The homologous chromosome pairs are separated and pulled to opposite poles of the cell. As a result, each cell is now haploid, which means it contains half the number of chromosomes as the original cell. The fourth stage of meiosis is telophase 1. During telophase, the nuclear membrane reforms and the chromosomes decondense. The cells also begin to divide in two. The process of meiosis then repeats again, but this time with 2 cells instead of 1. So prophase 2 is similar to prophase 1 in that new spindle fibers are formed. However, between telophase 1 and prophase 2, cytokinesis may or may not have occurred. Cytokinesis is the division of a cell into 2 separate daughter cells. The next stage is metaphase 2. This occurs perpendicular to metaphase 1. Individual chromosomes line up along the equator of the cell, once again attached to the spindle fibers by their centromeres. Independent assortment of chromatids occurs. After metaphase 2, anaphase 2 occurs. During this stage, the spindle fibers contract by breaking the centromere. This then pulls the chromatids to the opposite poles of the cell. Finally, telophase 2 occurs. Like telophase 1, the nuclear envelopes reform and the chromosomes decondense. Cytokinesis then occurs. The result of meiosis is 4 genetically non-identical cells, each with half the number of chromosomes as the original cell. As we have mentioned throughout this video, meiosis increases genetic variation. As a summary, this occurs during crossing over in prophase 1, independent assortment of chromosomes in metaphase 1, and independent assortment of chromatids in metaphase 2. To finish, here is a typical exam question about meiosis. It is worth 2 marks, so pause the video here and complete the question. It should take about 3 minutes. Okay, so here's the mark scheme for the question. To get the 2 marks, you should have mentioned independent assortment and crossing over. Then you should have explained that independent assortment gives rise to new chromosome combinations, or that crossing over involves swapping of sections of chromosomes. Thank you for watching. We hope this video has been helpful.